All right. Hello, everyone. Welcome to another session of our Evolution Seminar Series. Today, we have the last Early Career Award seminar of the semester. And for the last time this semester, this award is made possible by contributions from the College of Agriculture and Life Sciences, the College of Letters and Sciences, the Nelson Institute for Environmental Studies, and the Graduate School of UW-Madison. Really excited to introduce our speaker today, Takuji Usui. Completed his PhD at the Biodiversity Research Center at the University of British Columbia before joining the University of Toronto as a postdoctoral fellow. He's come to talk to us about his experimental evolution work on species ranges and species coexistence. So without further ado, please join me in welcoming Takuji Usui.
Hey. Hi, everyone. Yeah, thank you so much for coming to my talk, and thank you for your kind invitation. It's really an honor to be part of this seminar series, and I'm really excited to share with you all some of my recent experimental work looking at the evolution of species ranges and coexistence. So I consider myself to be a plant evolutionary ecologist, and as an ecologist, one of the things I'm most interested in is understanding the biological processes that shape species distributions and community structure across landscapes and over time. And this is, of course, a fundamental question of ecology, but also is an urgent question to address, of course, because as we all know, rapid changes in climate is leading to rapid changes in species distributions. So for example, here we have range expansions going on as populations spread across elevational gradients to track suitable climatic conditions, as well as range contractions at the trailing edge as populations may not be able to persist in situ to rapid warming. And along with these contracting and expanding species ranges, we also have changes in species interactions within communities as species kind of reshuffle and reassemble across the landscape. And I think one of the most interesting things to think about when we're dealing with contemporary species range dynamics is how these rapid ecological changes are also interacting with evolutionary processes. And then evolution is then also feeding back to further influence ecological dynamics in what we, of course, call eco-evolutionary feedbacks. And so today I want to talk about various ways in which eco-evolution feedbacks could kind of scale up to shape contemporary species ranges and community structure. And as many of you know, this is quite an empirically challenging and complex thing to study empirically, because first we have to deal with a whole bunch of ecological processes from individuals to community level processes. Then we're dealing with evolutionary processes. Third, we're dealing with its interaction. Fourth, ideally, we want to be able to manipulate or track eco-evolutionary processes across space and over time. And lastly, as an experimental biologist, I also want to be able to kind of follow these processes across replicate populations and across replicate landscapes. And so in my work, I use the experimental evolution of duckweed communities as model plant organisms in these kinds of large-scale mesocosm and lab experiments, and also experiments more recently in the field. And so duckweeds are extremely fast-growing flowering plants, aquatic flowering plants, with a generation time as fast as 2 to 3 days. I think one of the fastest-growing flowering plants. Experimental timescales, they grow mostly through asexual reproduction, through these daughter fronds that kind of bud off from their parental fronds, and also disperse away through breaking the stipule that connects the two fronds together. So I'll talk about these experiments in more detail, but hopefully I can convince you through the rest of my talk today that the utility of using such an approach to address kind of the fundamental ecological and evolutionary questions. So yeah, using this experimental system today, I want to talk about 3 different experiments in which eco-evolutionary dynamics alter species ranges and community structure. First, focusing on the dynamics with range expansion at the leading edge of a species distribution. Second, looking at range contraction. And then lastly, zooming back out a little bit and thinking about the evolution of coexistence and the evolution of biodiversity. So first, let's look at the eco-evolutionary dynamics of range expansion, and specifically how evolution could alter our ability to predict the speed at which populations are moving across space. So I really like to use this figure when we're talking about range expansions, just to really illustrate the point that as ecologists, we've spent a lot of time and effort trying to predict how fast populations are moving across space in response to climate change. And we found that range expansion speeds are extremely variable both within species and between species, and that they are really hard to predict. So although this figure is now a decade old, understanding what drives variation in range expansion speed and predicting the speed at which range expansion occurs for any given population or species is still a really large empirical challenge. And one of the challenges in predicting the speed at which populations move is that populations move across landscapes that vary in their selective environments across space. So even at the local scale, we have duckweed populations moving across urban gradients in, say, zinc or another pollutant. And as we all know, at larger scales, we have species and communities spreading across and straddling large-scale macroclimatic gradients in, say, temperature or precipitation. And so how does selection from these spatial abiotic gradients in space alter our ability to predict the speed at which range expansions are occurring? And can this explain some of the variability that we see in nature? So to test this question, we can first turn to theoretical models of theoretical population genetic models of range expansion. And first think about what our null expectation would be without any abiotic change, so across a uniform landscape. So here we have just a simple model of population spreading across space with the different colors denoting different genotypes. And then quite simply, the population genetic models predict that the speed at which populations move across space is governed by two parameters, so dispersal and growth rate. Quite simply, again, if you have higher dispersal or higher growth rates, you have on average faster range expansions. And these models, of course, also assume that these range-bound populations are typically characterized by low population densities, such that there is a strong effect of genetic drift, overwhelming selection. And so if we consider multiple replicate populations expanding across space, what this means then is that different replicate populations can randomly fix for different genetic variants in alleles. And for range expansion speed, which is what I'm interested in, what this means is that the chance fixation of different alleles can increase the distribution of possible outcomes, such that any single range expanding population can be harder to predict as we have a larger distribution of outcomes. So across uniform landscapes, then we have this fairly simple and intuitive prediction that drift should generate variation in range expansion speed, making range expansions harder to predict. So when we add an abiotic gradient, let's say from benign to an increasingly stressful condition, here theory predicts that if populations spread across space, there should be probably selection against dispersal and individuals also having lower growth rates as they spread into harsher environments. And this will lead to, on average, slower range expansions. And with this slow range expansion, then we may expect that populations at the range front to allow that these populations will have time to accumulate individuals at the range front, leading to high density population range fronts. And in these higher density range fronts, we would then predict selection to dominate over genetic drift and possibly favor a more consistent set of genotypes. So for example, if this was an abiotic gradient in temperature, perhaps this yellow and orange genotype confers some sort of thermotolerance. And then again, what this means for range expansion speed is that compared to a uniform landscape, we may see a more consistent set of outcomes across replicate populations, making range expansions less variable and hence more predictable. And so these are quite simple population genetic predictions, yet testing these empirically is quite difficult in nature because following and measuring the speed at which range expansions occur is quite difficult. Yes, so to test these predictions, I developed these experimental miniaturized landscapes at the UBC greenhouses in which I can experimentally track populations expanding across space from one end to the other. And so we had 36 of these replicate landscapes in total, which are made from these kind of rain gutters that are 10 feet long. And then to obtain our experimental founder population, I sourced different genotypes of duckweeds the common duckweed lemna from around the Pacific Northwest, and then seeded replicate mixed populations at one end of the landscape, and then allowed them to naturally disperse and grow across the landscape, across space, and over time. And then I was able to, using the system, track changes in population density, move how fast populations are moving across space, and track evolution happening by sampling individuals and tracking changes in the relative frequency of genotypes in the population. So importantly, half of these landscapes are spreading into across an abiotic gradient from benign to increasingly hotter and stressful conditions, while the other landscapes are spreading across uniform landscapes in temperature. So first, I wanted to look at the average speed of range expansion across these temperature treatments. So we have the distance traveled on the y-axis and time on the x-axis. So the speed would be just the slope of this graph. And we do find that in the presence of an abiotic gradient, so in gradient landscapes, the speed of range expansion is on average 50% slower. So that's great. It just means that our experimental treatment is working. Populations are moving into stressful conditions. So if populations are spreading slower into more harsh environments, does this then lead to higher population densities at the range front of these gradient landscapes? So here again, we have on the y-axis percent cover as a measure of population density. And we do see, again, consistent with predictions that across gradient landscapes, range front densities are much higher than across uniform landscapes, in fact, about 88% higher. So given that we have slower range expansions, higher population densities, do we then have selection or evidence for selection dominating over genetic drift in the presence of an abiotic gradient? And so when we compare average genotype frequencies across replicates in the gradient and uniform landscapes, immediately we kind of see this red genotype here, the G11 genotype popping out in this gradient landscapes, but not in the uniform ones. And when we look closer at the genotype frequencies of each genotype and just do a post hoc binomial test, we do see that this G11 genotype arises to higher frequencies than expected by chance in the presence of an abiotic gradient, but not in its absence. And when we look at the traits of these genotypes, we find that this genotype actually has the highest thermal tolerance or highest thermal breadth of any other genotype. And it actually fixes within 40% of replicates expanding across an abiotic gradient. And it doesn't fix in any of the uniform landscapes. So we do see evidence that there is genetic data that it's consistent with more selection going on in the presence of an abiotic gradient. So then what does this mean for our ability to predict range expansions? Is range expansions then more consistent, less variable, and more predictable across an abiotic gradient? So to look at this, we first estimated variability in the distance spread across uniform and across gradient landscapes as the coefficient of variation, and then calculated the log coefficient of variation ratio to look at the difference in variability. And so I've plotted that on the y-axis. So anything, basically, what the takeaway is that anything below that horizontally dotted line means that range expansions are less variable in gradient landscapes. So that's what we would predict. That's what theory would predict. So contrary to the prediction, we find that when we look at this variability over the time of a range expansion, we find that range expansion becomes increasingly more variable and less predictable in gradient landscapes compared to uniform landscapes, such that by day 24, range expansion speed is actually around 64% more variable, i.e. less predictable in the presence of an abiotic gradient. So what could be going on? Why, despite seeing higher population densities, potentially selection in abiotic gradients, why is range expansion more variable with selection in the presence of an abiotic gradient? So we have some data on what could be happening, but I'd like to leave this as a kind of an open question for now. And I'd be happy to hear your thoughts and ideas on what could be happening here. More generally, though, I think these kinds of experiments provide a nice tool to translate theory to empirical data and assess when deviations from theoretical predictions occur. And I just wanted to highlight that this is a very active area of research with a really nice crosstalk between theoretical models of range expansion and empirical data, with this really neat theoretical paper coming out just months after empirical data, showing similar things and slightly different outcomes. So in thinking about the eco-evolution dynamics of range expansions, I think both empirical and theoretical data suggest the need to more explicitly connect evolution dynamics to variability in range expansion speed in order to be able to predict what's going on in the future. So I think there are also a number of key gaps in predicting species range dynamics in the context of global change. So first, ecological niche models that we often use to predict future species distributions ignore these evolutionary dynamics, and I just explained that they might be important in driving at least the variability in range expansion speed. In addition, I think there are two more critical gaps. Just as crucially, I think we often fail to account for community context, and we know that community context and species interactions are really important in setting species distributions and range boundaries. So then how is this affecting the ability of populations and species to move across space with climate change? And then lastly, I would argue that in contrast to what's going on in the leading edge with range expansion, we know a lot less about what's happening at the trailing edge in terms of the dynamics of range contractions, even though these populations are experiencing some of the most extreme effects of climate change. So taking all these 3 points together, I'd like to next talk about the dynamics of range contraction, specifically thinking about how competition within communities affects adaptation at the range edge. Okay, so how does competition alter adaptation to warming at the range edge? So again, we can draw inspiration from theoretical models of evolution within a competitive context, and these models provide an array of possible outcomes. So one intuitive prediction is that the presence of a competitor reduces or constrains adaptation simply by lowering population sizes such that there is this amount of genetic variation that can respond to selection. On the other hand, though, other models suggest that competition could also actually promote adaptation. This could occur through something like shared trait responses. So a classic example might be something like root length. So if you have longer roots, you're a better competitor, but also might help you, say, in drought stress condition or under warming, because it allows you to also uptake resources more efficiently. And this additional selective pressure in the presence of a competitor then could promote adaptation in the context of climate change. And lastly, it might be that competition just doesn't matter at all under stress. So competition may not be important under stress because competitors are at lower densities or have weaker per capita effects under stress, such that competitive interactions are fairly weak relative to abiotic stress. And actually, what matters is that competition regulates population dynamics at the range core, where temperatures and environmental variation is more benign. So ideally to test these predictions, we really need is a scenario of range edge populations in the presence and absence of a competitor species, and also responding to climatic shifts across a species range from the range core to the range edge. So in our experiment, that's exactly what we did. So we tracked the eco-evolutionary response to temperature shifts in our focal lemna species in the yellow, with and without a competitor Spirodella species. So here's an area view of what our experimental setup looked like. So with our focal lemna species only at the top and our lemna and Spirodella communities at the bottom. And again, we have this temperature gradient going from the range core to fairly benign conditions to the range edge, which is more hot and stressful. And then we let the communities naturally grow and disperse across space and over time. And then when we established a species range under these conditions, we then shifted this environmental gradient across space, such that range edge populations were now experiencing increasingly hotter conditions. And then we let the communities naturally grow and evolve again in these new climatic conditions. And in total, we experimentally evolved these populations for around 2 months. After 2 months, then we conducted a full reciprocal transplant between the range core and the range edge, as well as between non-competitive and competitive landscapes. And by comparing the growth rates of our 288 transplant populations, we can then compare how populations with different evolutionary histories fare when we transplant them into different locations. So before we look at the evolutionary outcomes, first let's look at the demographic effects of competition across the range, both at the benign range core and at the range edge. So here we have population growth rates on the y-axis. And so we can first look at the range core, and we do see that competition reduces population growth rates, not a strong effect, but a reduction nonetheless of around 11%. Compared to range cores without competitors. When we look at the range edge, however, we see very little to no effect of competitors on population growth rates. So in terms of demographic effects, then this is more consistent to a scenario where competitors have a stronger effect under benign temperatures at the range core, but perhaps does not matter too much in regulating population dynamics at the stressful range edge. So then how does this effect of competitors on population growth rates then translate into evolutionary dynamics and adaptation? So here we have the magnitude of home advantage on the x-axis. So anything on the vertical dotted line means that home populations don't have an advantage. And as you move closer to your right, there's greater local adaptation in the home population. So focusing on the benign range core first, in landscapes where our focal laminar species are evolving alone without competitors, we see evidence of adaptation to benign temperatures at the range core, with significantly increased home advantage of range core populations compared to the other transplant populations. But interestingly, when we look at landscapes with interspecific competitors, we see that this evolved advantage completely disappears. So with a mean estimate falling right on the 0 line. So in other words, it seems that the range core competitors are limiting both population growth rates and the potential for adaptive evolution. Switching over to the stressful range edge, in contrast, here we don't see any adaptation to stressful temperatures at the range edge for laminar populations that did not have a competitor. But perhaps most surprisingly, in landscapes where our focal laminar species evolved with our interspecific competitor, we do see evidence for adaptation to temperature stress. In other words, at the range edge, while competition seems to have no effect on population growth rate, it still seems to be promoting adaptive evolution. And in fact, adaptation to stress can only happen in the presence of a competing species. So what's happening here? Why is a competitor promoting adaptation to warming range edges? So next, we looked deeper into what the traits that could be evolving in our populations. So here we have our PCA plot, where the PCA 2-axis is mostly explained by thermal breadth and critical temperature maximum, so the ability of populations to tolerate higher temperatures. So here I'm showing the evolved genotype-weighted means of these traits after experimental evolution in the non-competitive landscapes at the range core in the blue and at the range edge in the red. And when we plot our evolved populations that were experiencing competition, we see that competition kind of pushes these traits further up the PCA 2-axis, such that evolved populations has higher thermal breadth and also higher CT max. And so when we look at these traits in a more formal model, again, we see that this for both traits, for both CT max and thermal breadth, we see evolved differences between the range edge and the range core. So range core being in the blue and range edge being in the red. but only in the presence of a competitor species, and it doesn't happen in absence. So overall, competitors are enabling thermal adaptation at the warming range edge, and competition is strongly altering evolutionary trajectories despite having no clear demographic effects at the range edge. So we've been trying to think more about why this could be happening exactly at a more mechanistic level. And again, we would love to hear your thoughts on what could be happening. So to summarize this in a broader context, I think when thinking about how adaptation is unfolding at the warming range edge, we really do need to consider how thermal evolution occurs in the context of ecological communities. Maybe surprisingly, our results show that even antagonistic effects of competition does not necessarily lead to a more constrained evolutionary response. Okay. And so moving on to the last section of my talk, I'm going to change gears a little bit and zoom right out in scale and in perspective and talk about the evolution of coexistence and biodiversity over time. And so this experiment was a really fun collaboration between a bunch of community ecologists, duckweed experts, and evolutionary biologists. And in particular was also co-led by my friend and collaborator, Gerard Scarci, who is a PhD student with Rachel Germain at the University of British Columbia. So we've been talking about relatively simple two-species communities in experimental mesocosms, but in natural ponds, of course, we see an amazing diversity of species co-occurring together, from duckweeds to azollers and many species of algae and so on. And so understanding the origins and patterns of biodiversity is, of course, a key goal for many of us. But I'm going to argue that I think traditionally ecologists and evolutionary biologists have approached this question from quite distinct angles. And so to understand the origins and limits to species diversity, ecologists have traditionally focused on understanding the constraints on ecological differentiation and coexistence, and then how that limits species diversity. With Hutchinson asking in this seminal paper nearly 70 years ago, why are there so many species, arguing that niche availability ultimately allows for speciation, and limits to niche availability then constrains species diversity. And then, of course, 20 years later, Felsenstein came up and published this paper set up in direct contrast to the ecological perspective, arguing that coexistence only sets an upper bound to species richness at the macroecological scale. And what matters and what's important for determining species diversity is the microevolutionary processes controlling the evolution of reproductive isolation between genetically diverging populations. And so as someone who thinks about eco-evolutionary feedbacks and eco-evolutionary dynamics, I kind of wanted to try and bridge these two perspectives together, bring the community ecological and micro-evolutionary perspectives on the origins of biodiversity. And so what do I mean by that? So first I want to talk about how and when, relative to the evolution of reproductive isolation, do we see ecological differences evolving among nascent lineages during speciation? And secondly, do these ecological differences that evolve lead to the evolution of lineage coexistence during speciation? So before I talk about why bridging this gap and answering these questions could be important, I want to give a quick overview on what I mean by ecological differences and coexistence. So, so by ecological differences, modern coexistence theory states that there are two modes of ecological differences: competitive differences on the y-axis there, and niche differences on the x-axis. And coexistence is a balance between these two modes of ecological differentiation, such that if you increase niche differences between populations of species, you increase your probability of coexistence. But coexistence can also be increased by decreasing competitive differences. So if two competing species have more similar competitive ability, they're more likely to coexist together. So why do these? Let's think about how these different modes of ecological differentiation could evolve among nascent lineages And why is this important when thinking about species diversity and speciation? So I think let's consider first the case where we see the rapid evolution of, for example, niche differences between diverging populations. So if rapid niche evolution actually happens before during the niche divergence, but before the evolution of reproductive isolation, let's say between allopatric populations that are diverging, genetically diverging, when these lineages come into secondary contact, then this may lead to lineage collapse because coexistence is allowing these populations to co-occur together and potentially hybridize, leading to the collapse of this diverging lineages. On the other hand, we may see that coexistence between lineages actually allows speciation to be successful if niche differences actually evolve after the evolution of reproductive isolation, such that when those lineages then come together in secondary contact, they are able to persist across the landscape. And then on the other hand, if we see competitive differences evolving during lineage divergence, then we may see competitive exclusion of one lineage over the other, leading to unsuccessful speciation, because one of the diverging lineages be able to competitively exclude the other diverging lineage when they come into secondary contact. So to answer this question more empirically, we used contemporary populations of the giant duckweed Spiraldella polyrhiza. And so for our experiment, our collaborator Shuking Xu sampled 126 unique populations of Spiraldella polyrhiza, with each lineage coming from different points around the globe. And then for our experiment, we actually simulated secondary contact between each of these allopatrically diverging populations to test the ability of these lineages to coexist upon secondary contact. So we conducted, this is what the experiment looked like, where we conducted reciprocal pairwise competition experiments across 442 unique combinations of Spiraldella populations in these buckets in the greenhouse. And this is what it looked like inside each bucket, where we have a resident population in the background as the resident genotype, and then we have an invader population coming in to each population. And then we are essentially testing the ability of each of these invading genotypes to be able to coexist and grow from where. So usually these kinds of coexistence experiments are done at the species scale. So we are able to tell apart which is the resident and invading species. But in our case, we're computing two populations of the same species. So we actually had to manually track and hand count each individual duckweed growing in the context of the background competitor. And we did this by hand spotting each individual lineage across 8 days, tracking them by hand by putting a small white dot and then seeing how they grow in the context of a competitor species. And so we actually also have whole genome sequences from each of these allopatric populations, so that we can estimate coexistence across genetically divergent lineages and test how these mechanisms of coexistence are accumulating across genetic distance. So first, do ecological differences actually accumulate among nascent lineages? And so here we have two modes of ecological coexistence, so niche differences on the y-axis and comparative difference along genetic distance from identical populations to further diverging populations. So we do find that niche differences significantly increase across genetic distance. Our data is fairly noisy, but we do find that regardless of how we model this data, there is a linear trend or a non-linear trend that increases niche difference across genetic distance. On the other hand, competitive differences don't seem to change across the genetic distance. And when we plot these two ecological differentiation modes back on our coexistence plot here, we see that most population pairs fall within the coexistence zone. So most populations fall within this shaded region here. And so when we extract this coexistence data and then plot this across genetic distance, so we're asking what is the probability of coexistence that these diverging lineages are able to coexist together as they genetically diverge. So we also find that the probability of coexistence increases across nascent lineages. And so it's interesting because we usually think about coexistence accumulating across phylogenetic scales between species, but we actually find that the evolutionary relatedness between populations within species also predicts the level of coexistence. And so we find for the first time that ecological coexistence mechanisms can accumulate at timescales critical to speciation. And this is important because the fact that mechanisms of coexistence between nascent lineages can accumulate at these timescales means then that the evolution of coexistence mechanisms have the ability to kind of potentially interact with the evolution of reproductive isolation. And then this eco-evolutionary feedbacks can then determine the outcome of successful speciation. Okay, am I running out of time? 105. It goes to 110.
130.
130. Okay. Oh, perfect. Okay. Yeah. So and again, this point was actually made also in a recent theoretical papers, trying to reconcile these two diverging perspectives of ecological and evolutionary perspectives on diversity, saying that the evolution of both reproductive isolation and coexistence together can constrain the rate of speciation and diversification. And importantly, I think this neat perspective piece came out as well on the variable success of linking microevolutionary dynamics to macroevolution, suggesting that this potential ecological limits controlling speciation could explain this missing link between microevolution and macroevolution. So then we became also interested in this idea of how these population level processes predict patterns of coexistence across larger phylogenetic scales. So we have data right now of the probability of coexistence within populations across genetic distance. But what if we extend this idea to beyond the species boundary? So to investigate this, we actually also parameterized coexistence across the species boundary at the sister species level, doing the same kind of reciprocal pairwise competition experiments, but this time between each of our Spirodella polyrhiza genotypes and population of its sister species intermedia. And so going back to our coexistence plot here, this is the data that I showed you of coexistence within species. And then we can add our estimates of that we measured of coexistence across species. And we see that niche differences continue to accumulate across at phylogenetic scales, allowing further coexistence of at the species level. And then going back to our plot of niche differences across genetic distance, when we add our estimates here across species, we find that niche differences do accumulate across the species boundary, but that this accumulation is actually nonlinear over phylogenetic timescales. And so if we actually try to estimate the level of niche differences from within population data to across species, it's actually around 51% less than predicted from a linear model of within species data. So we find that niche differences increase at phylogenetic scales, but opportunities for niche differences saturated quickly at the species level. And we're trying to think about what this implies and what this means in terms of diversification and speciation rates at a macroevolutionary scale, and linking those population level processes to larger macroevolutionary timescales. Okay, so overall, I would argue that the evolution of coexistence can happen really quickly between diverging populations, and that this should interact with mechanisms of reproductive isolation to then determine the evolution trajectory of speciation. So I just wanted to finish quickly by highlighting duckweeds as a model system and some of the ways in which I'm using them right now. And so, so I think duckweeds are an excellent model for studying plant invasions and understanding what makes plant species that are so globally distributed and really so successful at colonizing new habitats. And so right now we have around over 200 separate lineages and populations of lemnos species from across North America, with collaborators coming in with some population genomic data. And we're hoping to actually understand the population history and the demographic history of duckweeds in North America. For example, how many colonization events there were, where they're coming from, what's actually happening in field sites. And then one of the interesting things we've also found was that population genetic and population genomic diversity within ponds and even at the local scale between ponds is extremely low. And we actually only find only one genotype and one species of duckweeds often occurring in between one pond. But our data earlier showed that actually many genotypes of duckweeds are able to coexist together, and many species are also able to coexist together. So why is this not happening in natural ponds? Why is, despite there being frequent migration events between ponds, transferring duckweed populations and mixing them around, why do we only really see one clonal population at a given time? And then another kind of angle I'm exploring is using duckweeds as an experimental co-evolution model of host symbionts. So actually the microbiomes of duckweeds have been characterized for a long time, since the 17th century. And we're able to do manipulative experiments where we have entirely axenic and sterile plants. And in well-played experiments such as this, we can then inoculate different microbial combinations in different microbial communities and really study the effects of co-evolution between plants and their microbiomes. And then lastly, I'm also trying to develop these duckweed systems for more high throughput and semi-automated experimentation and phenotyping. And so we have these handling robots where we can do set up semi-automated experiments. But also developing more software using computer visions, where we can actually track individual lineages of duckweed growing within experimental timescales. And then so we are able to actually create lineage trees of different genotypes within the population. And so I recently got this data back where we have genotype frequency on the y-axis. And this is 180 replicate populations and different genotypes just sorting across over time within an experiment. So then this is quite powerful because we no longer have to do actually extracted sequence information. We can actually visually monitor how evolution is unfolding. Okay, so yeah, thank you again for your invitation, and I'll take any questions. Yep.
So where you found a improved adaptation to thermal stress in populations that experience competition. So the conventional wisdom is that if you throw many selective pressures at an organism, their ability to adapt is reduced to factors like antagonistic pleiotropic total interference and so forth. Why do you think you're seeing this result that runs so much counter to the theoretical predictions of conventional wisdom on the subject? I think what you're saying is not generally true, and you think that multi-drug cocktails will not be effective against viruses because they have antagonistic selective pressures, and yet we know that multi-drug cocktails are effective.
Yeah. So why am I seeing? Why do you think I'm seeing this effect of competitors actually promoting adaptation? When we expect, when we have multiple selective pressures, then theory predicts otherwise. Yeah, so that's something that we're really trying to narrow down and pin down. One kind of data that we do have is on we think that the presence of competitors is actually changing how the focal species lemna is experiencing selective pressures from the environment. So we know that those competitor species can form these kind of large mats in the landscapes, and potentially creating small kind of like microhabitat differences in temperature and changing actually the selection that is being experienced by the focal species. Maybe I have to on that. No, I don't, sorry. But yeah, that's one idea that we have. The other idea is that these small differences in microhabitats are actually allowing population sizes to be maintained at a low level, so that the actually genetic variation is able to persist in the landscape, but can't without this competitor. So effectively increasing environmental heterogeneity in the landscape. Yeah. Yeah.
I'm curious about the paradox of duckweed and the arguments about coexistence and competition.
Yeah.
They never experience, or at least rarely have, like intense competition from what we can see with a single genotype in a single pond. Does it make it a better or perhaps worse model for arguing that they're reconciled those two?
Yeah, no, it's a good question. Yeah, so we don't, yeah, I think just generally we don't know, we don't know how actually how much they're competing in nature in these ponds. So we don't have good space. I don't think we have good enough spatial resolution to say whether competition is actually occurring between different genotypes within the pond and how much that's actually occurring. Yeah, I mean, sorry. Yeah.
Like if you found some where there was actually a few genotypes in the pond and asked whether they were outliers, perhaps in your competition niche threat models or something like that.
Yeah.
There's no evidence that even under competition, there's something popping out differently.
Yeah, no, actually, so yeah, our populations actually include genotypes that have so that naturally co-occur with other species and also don't. And yeah, it doesn't, yeah, it's we see some differences in competitive ability between those populations, but when we look at the coexistence plot of competitive ability and niche differences, it doesn't seem to really pop out in any way. Yeah. Yep.
Yeah, there is an explanation for this idea that you get more evolution in the presence of a competitor. Just really quickly, two background pieces of information. You can't think about this as a tree competition system, right? I mean, they're clonal, so there's no recombination, so
Mhm.
three competitors. Second thing is there's a paper by Chesson and Huntley actually. This shows that in really extreme harsh environments, it doesn't mean that competition is any less.
Yeah.
It's neutral. I mean, in a harsh environment,
Yeah.
sure, the per capita effect of the total effect of competition is less because the population
Yeah.
reduced, but per capita effects are more strongly.
Yeah.
Right?
Yeah.
So there's this kind of trade-off between abundance and per capita effect.
Yeah.
In one particular situation, thinking about it in terms of three species, then you could have one genotype that does better at high temperatures, the other one that does better at low temperatures. There's also this trade-off in terms of overall population growth rates, which depend upon the third species, right? So it's a kind of balancing act between the two.
Mhm.
And all the competition is doing is changing the relative importance of the per capita population growth rate due to the intraspecific interactions versus the difference between the two genotypes in terms of environmental stress.
Mm.
Sorry, this is a little bit ornate.
Yeah, no,
We can
yeah.
map it out in more detail tomorrow, but we're going to breakfast together.
Yeah, sounds great, yeah.
Yeah, I mean, it actually, if you kind of think about it, it's an ecological problem.
Yeah,
There's antecedents of stuff like this.
yeah, cool, thank you, yeah. Yeah,
Just curious, in the process of testing these kinds of dynamics, is there any reason to think that some of this might be trait specific, depending on, like, you're analyzing heat, obviously for its connections to climate change,
Yeah.
but could some of these dynamics be trait specific? Like if you looked at range expansion into higher salt,
mhm,
like, for example, heat is such a broad stress
Yeah.
on the cell that you can imagine there's multiple avenues.
yeah,
And that could potentially explain that higher variability you saw
yeah,
that was counter to the theoretical prediction. So I'm just curious
yeah.
if have you thought about testing other traits and how that might affect some of these dynamics?
Yeah, that's a good question. Yeah, I think I'm really kind of getting more curious into thinking about this from a more physiological perspective. So, like, yeah, yeah, I think, on traits, it's kind of hard to measure different traits in duckweed because it's so simple. But yeah, so I'd like to kind of incorporate a more physiological or even a little bit more metabolic perspectives to see how these individual differences are scaling up to population differences, and so we can kind of work around so that it doesn't become so trait centric, and there are maybe underlying processes based in physiology or metabolism that we can scale up to understand different abiotic drivers. Yeah.
I had two questions. One which is just a clarification on my part, and another which is all the way back to differences in variation as they increase.
Oh yeah.
The first is, would you go quickly through again how you got from in the buckets of duckweed, you're doing competition, how you got to from the phenotypes of the counts of duckweed to the actual niche differentiation and competitive value?
Oh, and the coexistence part. Yeah. So I actually skipped that because I was panicking about running over time. But so basically what we do is so coexistence theory predicts that the way you parameterize coexistence is that you invade an already established resident population. So it's pairwise, which is limiting, but so you have one lineage and the second lineage at their carrying capacities, and then you introduce a competing lineage, and then if they can invade from low densities, then they can coexist. That's how coexistence is parameterized. And then the part about the niche and fitness differences comes from calculating what we call the sensitivity in growth rates. So basically you compare the growth rates of an invader alone versus when it's growing with a competitor species, and then basically calculate differences in the growth rates with and without a competitor. So that's the SI term that becomes the sensitivity in growth rates to competition. And then from that, there are different ways to calculate niche and fitness differences, but then one way to calculate is using these sensitivities. So niche differences is actually just the the one minus the geometric mean of the sensitivities. And then competitive differences captures the variance in competitive ability between the two competing genotypes. So if you have lower on average sensitivities, you have higher niche difference, etc. Yeah.
Thanks.
Cool.
The suggestion on the variation in growth rates early on, it reminded me a little bit of, like, if there's a period in adaptation if the variants that are adaptive are very low, in which it's in drift dominated dynamics.
Yeah.
And the wait time to escape from that period is a random variable, but the growth rate is somewhat deterministically set after that. So if once a significant portion of the population has the adaptive genotype, the rate of growth and the rate of adaptation will both be mostly deterministic.
but the wait time is much more variable.
Yeah.
And so you might think that maybe that's an explanatory variable that gives you such variation. Are you?
Yeah,
based
yeah,
on that,
yeah.
Mostly it's the timing at which they are at maximal growth rate that's varying between these replicates.
Yeah, that's a good idea. Yeah.
It's kind of similar to if you take like bacteria out of like starvation. Like if you're taking them out of freezing or starvation, there's a random variable as to when they start growing. That's just this,
Right.
and that generates a large amount of variation in the initial genetic variation. In
Yeah.
samples.
Yeah. Cool. Yeah, that's a neat idea.
All right, let's all give Takuji a big hand.
Yeah.